this is indeed me. And my defining quality is that I'm not a geneticist, which you will come to see. So important disclaimer before we begin. What I'm going to show you is a very preliminary data. So I'm mostly here to show the experiment design and share the research questions. So treat it as a trailer for future talk. I've been informed I'm allowed to do this. And if not, I'm prepared to face your wrath. Okay, so it is well established that mitochondrial function declines with aging. However, the precise relationship between mitochondrial dysfunction and aging is not clear. In particular, it is not clear what exactly mechanistically goes wrong with mitochondrial machinery. We have a lot of reports, a lot of data, but it is buried. Additionally, one of the reasons it is so hard to establish what goes wrong with mitochondria with aging is because any trait concerning dealing with mitochondria, having to deal with mitochondria, is a especially complex trait. So it's actually, I would argue, it's as complex as it gets because there are two genomes involved, and for the core mitochondrial machinery to work, these both genomes must work in unison. Additionally, while nuclear changes to the nuclear genome are turn-based, so from generation to generation, nuclear genome it works in real time, so it changes all the time, replicates, mutates all the time. Additionally, these dynamics might be different in different tissues, which adds to the complexity. We don't really understand the relationship, like the causal coupling between mitochondrial dysfunction, aging, and AD, what drives what, in what order, and as usual, how these all interact with genetic makeup. So for this research, we're going to use murine models. So the prediction factors, the grouping factors, are going to be sex, which is implicated in aging. The age group we use are, as you can see, 6 and 14 months. The one is like it's mature, so it's mature adult. It's finished. It's finished its maturation, but we don't expect it to have any age-associated conditions. While in the 14 months, we do actually expect. We do hope to find them. The strains are like B6s as controls. We're going to use BXD. I don't need to tell you what BXD is, but importantly, BXD we use because it provides us with replicable genetic diversity, which harkens back to the aforementioned genetic makeup. So we use like long-lived BXD51 strain, and we also scheduled BXD60 as a more short-lived, and importantly, it has alterations. Okay, it's actually the next point. So the next point that all our strains are also crossed with 5X BXD to provide for Alzheimer's disease like phenotype. So, and this is where like BXD60 works, as Joy mentioned. Yes, so here you can actually appreciate how much this genetic diverse genetic makeup affects like supposedly very straightforward transgene. All right. So, and it is found in humans, but not in many animal models. Okay. So a lot of work has already been done by my coworkers working with mitochondrial DNA copy number, and with it, we do in fact see a pretty good correlation of lifespan and DNA copy number. So this is like promising data, but this provides some, in terms of prediction and intervention. But when it comes to prevention specifically, we will need to get to the functional level. No, I'm gonna yeah that was liver, yeah, right. So for the thing we're going to do for functional analysis is called respirometry, high resolution respirometry. And so as you can see, we have a lot of combinations, and we would also like to see different tissues. However, this is a very low throughput method, and we need to be very conservative with what we can measure, because normally you would like with respirometry, normally you would like to sample at least 5 to 8 animals per group, and basically measuring just one tissue takes half of the day. So this power law sends us right to the next decade, and I would like to finish this experiment before I reach human endpoint. Yes. So the thing we were trying to do to go around it is, instead of just adding groups upon groups with any factor, we just use basically nested design. We take a bunch of animals of different combinations of these factors, and we kind of rotate this like we can, in the same bunch of animals, right? We consider each grouping factor separately. So first we just see young versus old. Then we do like non-transgenic versus transgenic. Probably can see it, but the lining is different. Should have been larger grain. Okay. So, and then we intend to integrate basically, yeah, we intend to run a linear mixed model to see what contribution each of these factors makes, right? And for the actual, boots on the ground protocol, this is what we're going to use. This is the essence of mitochondrial respirometry. So we basically isolate mitochondria. We add them to a sealed chamber, and the thing this machine actually measures is oxygen concentration, oxygen dissolved in the like medium in the chamber. And if mitochondrial respiratory chain works at any capacity at all, it starts reducing oxygen. However, the condition is so designed that first, this chain does not function at all. So we can turn it on little by little, step by step. So first we add substrates, but no ADP. So ATP synthase is stalled. We have something called proton leak. So it's inevitable, but this value could vary depending on how well mitochondria are made. So it's basically, as it says on the tin, some protons just leak through the membrane, or there's some slippage of proton pumps. Then we actually add ADP and let the protons in. That's oxphos. This is like normal, what's normally happening in the body when mitochondria make ATP. And then we just completely uncouple it. So we make membrane permeable to protons, so that respiratory complexes work at maximum speed, at maximum capacity. So it provides us when we subtract OXPHOS from this parameter, we have spare capacity. So what mitochondria have on top of usual OXPHOS, so like compensatory, capacity. Okay, so we can also selectively turn off certain complexes to see it more selectively. Right, so like complex 3 plus complex 4 just separately. And this is the preliminary of data we get. So, it's often advised against to look at the data amid experiment because it might bias you. So I looked at it very carefully with safety screens engaged. So, and these the RSR, it's like an integrative parameter. Sometimes it's used as a like quality control, but also it just provides you an integrative measurement of how well mitochondria works. So this it's not fit to do proper statistics so far, but the general look of the numbers makes sense here. This we had half decent number of points for the liver. So the numbers are within what we would expect. So no anomalies here as well. And basically we have some data for mitochondrial DNA copy number. It makes sense. Spirometry pipeline established, and data collection is well underway. And I anticipate these points of data to couple and start making all kinds of senses. Many thanks to my co-workers from David's lab and David personally, and also Dr. Olwe and Hector, whose advice and expertise were instrumental in setting up the respirometry pipeline. Thanks so much. I'm prepared to answer your questions now.